about really the structure of nervous tissue and looking at neurons and glia. And today we're going to continue on with the chapter on nervous tissue, and we're going to start to look at the function of nervous tissue. And whoops, wrong picture. Yeah, okay, so we ended last time looking at how myelin forms around axons. And now we're going to look at how these cells that we're talking about interact together to produce the nervous system function. And we'll be looking at starting to learn about the electrical signaling aspects of nervous tissue so that we can understand how the communication and the pathway like this takes place. Now, I think I said earlier, the book that we're dealing with here, which I actually wrote these chapters, is in its first edition, which means that it has shortcomings. And personally, I think one of the biggest shortcomings is in how I present this material in this part of the chapter. So I'll explain things based on how I have it in the book to some extent, but I'm trying to modify it and make it more straightforward. So we'll work on that to somewhat through this. Now, one of the problems I have with it is actually what this figure is showing. The point of this figure is to demonstrate how how different components in the nervous system work together for a complex behavior. And a book I used to use has a similar sort of picture, and I can't remember now exactly what the picture demonstrates, but it's a more probably straightforward example of what I'm trying to get at here. And so basically working with the editors of the book, we needed to come up with something that was unique, I suppose you could say, and not sort of step on any other publisher's toes and copy their work. So I came up with this idea, which actually in retrospect I don't really like. It came from actually something that I do think illustrates the idea at a simple level pretty well, which is the reflex, the withdrawal reflex, that when you touch a hot object like the stovetop, you withdraw the hand. Okay? And the problem though with that is that that's a very simple circuit. There's really only 2 cells involved in that. And what I wanted to do was to include some central processing to show how the sensory and motor output, the sensory input and motor output of a system like this interacts with higher functioning to generate behavior. So I took that touching hot stove idea and went this way, which in retrospect I don't think is the greatest example. But what we're seeing here is a hand that is under shower water to check the temperature. And so there are sensory endings in the skin, which don't necessarily have to be in the fingertip. They're going to be anywhere in the skin that the water's falling on, that are sensitive to temperature. Okay, so the temperature of your body should be 37 degrees Celsius. And if you expose the skin to something higher or lower in temperature, then there are sensory endings, dendrites of unipolar neurons, which will be activated by a difference in temperature. That stimulus will cause the generation of an electrical signal called an action potential to travel along the axon in this peripheral nerve through the hand and the arm up to the shoulder and into the neck region, where the dorsal root of the spinal nerve connects to the spinal cord. In that dorsal root, we have that ganglion that I showed you in lab, where the cell body is located. But the axon continues on into the spinal cord and turns into a region of white matter that's right here in the dorsal part of the spinal cord. It goes up the spinal cord and eventually heads to this part of the brain right here, where it finally makes a synapse. Wait a second. Oh, sorry. I missed the synapse. It goes to this point right here and makes a synapse. Now, the picture of the brain we have here does not have the brainstem in it because we need to have a picture of this stuff here. So this thing that seems to be floating around outside the brain is actually in part of the brain, the base of the brain brainstem that connects to the spinal cord. So this axon, starting in the hand, goes all the way up into the brainstem and makes a synapse on a target neuron there, carrying the information about the temperature of the water to the brain. And then from the brainstem, there's in the brainstem, there's a nucleus that contains neurons, which are the targets of these sensory neurons. And they send information along to the next stop, which is another nucleus in the brain that contains more neurons that are targets of this second neuron here. That target neuron, the third one in the line that we're looking at here, when it receives input from the second neuron, it will send a signal on up into the higher centers of the brain. This specifically called the cerebral cortex. Now, in this picture, because it would just get way too complex if I try to be as anatomically correct as possible, there's not a neuron in that at the end of that third axon acting as a target in the picture. There is, but it's not drawn in here because at that point the circuit gets way too complex for the sake of what we're trying to talk about here. So this just represents all three of these blue colored neurons are carrying the sensory information into the brain. And so when it gets up there to the cerebral cortex area, that represents where the conscious perception of the temperature of the water takes place. The time it takes for it to get all the way up there, which is, in terms of cellular size, very long distance, can be measured in hundreds of milliseconds, if even that much. And so from when you actually put your hand into the stream of water to when you register what that temperature of the water is a fraction of a second. So you wouldn't necessarily perceive the delay between activating the sensory receptors in the fingers to when you actually sense it up there. But there is a little bit of delay representing the movement of the electrical signal on up to the brain. One problem with this picture, which I didn't even notice until after the book was published, made available, whatever you want to call it, the spinal cord and the brainstem should be lined up. So this is the midline of the spinal cord, this is the midline of the brainstem. They really should be lined up to be as accurately, anatomically accurate as possible, because where the blue line we were just following and the red line I'm about to talk about sort of turn here, that's just a weird little quirk in the drawing, because they would go straight up from the spinal cord to the brainstem or straight down from the brain to the spinal cord. This is not anatomically accurate, but it's necessary for the way that this picture was laid out. And the reason why they did that was just all this stuff at the bottom, if it lined up with the brain, would have to be shifted over, a bit. And the picture would be this wide over here, and there'd be a lot of seemingly empty white space over there. Graphically, I understand why the choice was made. It actually intuitively, as a graphic design choice, made sense to me, and I didn't even think to say anything about it when we were putting the picture together. But in retrospect, from an academic anatomical point of view, that was really a bad choice. Okay. Just that you really should see that from the spinal cord up to the brain is actually a straight shot. There's no turn in the middle of things. And also because of that turn, the way they drawn it, you see the red and the blue crossing over here and crossing over here, which suggests that the two pathways kind of go back and forth, which is not true, and it's just a quirk of the way the things go. So, I would fix that. Really what I want to do is I want to fix the whole thing and come up with a completely different example, but I haven't figured out what example would do better yet. So, anyways, I was describing the sensory pathway that's coming in. There are 3 neurons that are part of that pathway. The one that's actually interacting with the stimulus, with its sensory endings in the finger, is the sensory neuron. And that's labeled right here. Okay? When we talk about the location of a neuron, of a particular cell, implied in that is where the cell body is located. Okay? So if I ask you where is this sensory neuron located, the answer would be in the dorsal root ganglion. Okay? Its dendrites are in the skin, its axon synapses in the brainstem. So it spans a very wide region of the body. But the center of the cell, where the nucleus is, where the cell body is in that ganglion. So that's considered that neuron's location. That's why it's labeled right there. But this is a sensory neuron, which means usually that it's the one that's directly getting the sensory stimulus. It then synapses onto this cell, which is labeled as an interneuron, which just means any neuron that receives information from one neuron and passes information on to another neuron. It's in between 2 neurons. And it synapses on this, which goes up, which is also labeled an interneuron because it fits that definition that it receives information from one neuron and is passing it on to another one, which isn't drawn in the picture, but does exist. Now, they're all colored blue because they represent a sensory pathway. Now, I said sensory neuron usually is defining the neuron that's getting the sensory input directly. But technically, or I shouldn't say technically, but under some circumstances, we use sensory neuron to apply to any neuron that carries sensory information. So those 2 interneurons that are also colored blue are sensory because they're part of a sensory pathway. If we isolated that neuron by itself, it's not getting any sensory information directly, but it's part of a pathway that's carrying that. So more often you'll hear sensory neuron describing the neuron that's actually interacting with the water stimulus. But if you ever hear sensory neuron applying to something that's technically an interneuron, just understand it's functionally carrying sensory information. Okay. Now, what's left out in the picture, because I said it would just get too complex, is in the cortex we have neurons that receive information from the sensory input. They process that information, that integrative function that I mentioned last time. I did mention that, right? Integrative. I don't remember that. I had mentioned it. I'm sure I did. I just hesitated a little bit because of your lecture, and then right after it, I did another lecture, and I definitely remember writing it up on the board for them. But I'm pretty sure I did. It's in the book, if I didn't say it. Just where it talks about sensory and motor function, it also talks about integration as a complementary function. But integration means that we can't say that it is sensory or motor exactly. Okay? It's putting information together to arrive at a decision point for what the response is going to be. And so in what seems like a very short distance right here, there's going to be a whole circuit of neurons involved in that processing, that integration, which will eventually end by sending a signal to this cell right here, which is also in the cortex, just in a different part of the cortex. And in lab next week, we're actually talk about the areas of cortex, defining what they do and where they are. So I'll come back to this and actually say this part of the cortex does such and such, and this part of the cortex does such and such, and you'll see why they're next to each other, but they're distinct regions. The cell here in this part of the cortex is now colored red to represent that it's for motor function. And its axon descends down through the white matter of the brain through the brainstem and into the spinal cord, where it finally synapses on a second motor neuron in the ventral horn of the gray matter of the spinal cord. Okay. When we were looking at multipolar neurons in lab this past time, that first region that we looked at, the first cells that I showed you, were these cells right here. Okay, that's the part of the spinal cord we were looking at. And their axons extend from the gray matter of the spinal cord through the ventral root of the nerve, and then the nerve carries it through the shoulder, the arm, and the hand, and it eventually synapses, makes neuromuscular junctions with muscle fibers in this example, the hand. Okay. So one of the problems with what I'm doing here is the reason why it's not a great example is this is just showing the sensory input and the response in the same hand. But if you think about if you were actually testing the water in the shower, you would put one hand in the stream of water and use the other hand to adjust the temperature or something like that. And there's another picture of a hand adjusting one of these. So it didn't quite work out the way I was hoping. So this is saying that in response to what that temperature was with the integrative processes that aren't drawn in, it'll eventually cause a response by causing contraction of the skeletal muscles in the hand, and the hand will do something, which might be pull out of the water. If you put your hand under there and it's too cold and you're like, oh my God, it's too early in the morning for a cold shower, you're going to quickly pull the hand out of the drinking water. Whether that hand or your other hand actually adjusts the temperature of the water isn't important, but that's kind of the idea. The point of this example was to show the entire pathway from receiving sensory input and to producing a motor output. And in retrospect, it's just not the greatest example for that. I need to think up a better one. And unfortunately, a lot of the good ones are already used by other textbooks, and we needed something unique and not the best idea that I ever came up with. There's another problem with this picture that I want to point out, which is the representation of sidedness in this picture. So the sensory input to the cortex and the motor output from the cortex are shown coming from the left side of the brain. And they are going to be connected to the right side of the body, which you see, and eventually they come out, or go in or come out through the right spinal nerve at that level of the spinal cord, which is correct. Two problems with that in this picture. One, having to do with the fact that the spinal cord and the brain didn't get lined up on the same midline, is that the drawing shows, of course, the red and blue lines sort of jogging off to the left side of the picture. Actually, what would happen is that they cross midline to go down on the right side. The left side of your brain connects to the right side of your body. And that fact is just lost in this weird quirk that I mentioned before because of not lining up the brain and spinal cord. The other problem is that this is not the right hand. There's right hand. If we were looking at the right hand in this point of view, the thumb would be in the back of the picture, but it's actually in front. So for whatever reason, they drew a left hand, but said that it's the right hand. I mean, they don't say this is the right hand, but from all the other aspects of the picture, that necessarily has to be the right hand. Just not the right hand that they drew. So a lot of students probably wouldn't pick up on that little nuance, but I did want to make a point. In case you're looking at that and you're saying, well, there's my hand, and you're like, wait a second, my left hand would be connected to the right side of my brain, but up there it's strong. So just understand that's actually a mistake. But that sidedness is important. Sensory information that is received by nerve endings in your skin on the right side of your body initially goes to the left side of your brain. And that's where you start forming conscious perceptions of what's happening on that side of your body. Excuse me. And then any voluntary command to move muscles is going to originate from the opposite side of the brain. So the left side of my brain is telling me to move my right arm like this. The reason for that is not entirely well thought out or explained, I suppose you could say. I actually one of my classes in graduate school was specifically sensory neurobiology, which is sort of measuring or talking about what's receiving stimuli and sending that into the brain. And the professor there, who actually kind of scarily enough, I emulate a little bit more than I ever thought I would, because he was a weird guy. Nobody? He posed the question to us of why does nervous system connections cross midline? And he was not saying that there was, but the way that he asked the question was sort of implying, is there some sort of cosmic reason for all of this? And so we thought about it, we discussed it. Interestingly, the research that I was doing in the lab, I think actually gets at the reason for this, which is not at all cosmic or anything like that. It just happens to be a sort of mundane aspect of how nervous tissue grows, which is this axon here, for instance, from the motor portion of the cortex, grows down from a very anterior location of the brain. And I'll explain why we call that anterior in lab next time. Deeper and deeper into the brain, and eventually straight down into the spinal cord, which is more posterior parts of the nervous system. And again, I'll explain why I'm using anterior and posterior later on. But that direction of growth, anterior to posterior, has to match with a medial lateral growth as it, or that doesn't matter. It ends up matching with the medial lateral growth as it's growing down from the brain to the spinal cord. And the reason for that is that one of the signals that tells the axon how to grow in the embryo comes from midline. And so an axon is growing along and it's getting that signal as it approaches midline. And when it gets a certain strength of signal, it actually turns and crosses over to make sure that it's growing in the right position. So it's actually a little chemical signaling quirk that makes things cross over from one side to the other. There's actually not any real cosmic reason that that should be. It's not like people got damage on the right side of their brain more often than the left side of the brain, so the left side of the brain was in charge of the dominant side of the body or something like that. It's not along those lines. It's probably just because of the complexity of making these little cellular components like axons grow very, very long distances. That they have to get to the decision points, and sometimes the decision is turn left at Albuquerque. And because of anybody know that reference, Bugs Bunny? All right. But they get to a certain point, they have to make a turn, and that turn happens to send them across midline. So the left side connects to the right side. And it seems to be a very fundamental part of the nervous system, and I just want to take a little bit of time to explain that because I think it really is just a matter of how the complexity of the nervous system has to develop in the embryo. Now, the other thing that I want to point out here, or actually there's a couple of things, and before I get to what I was about to say, let me say the second thing, which is in the motor pathway, the two red neurons here, they're labeled as the upper motor neuron and the lower motor neuron, which hopefully is kind of straightforward idea. The upper motor neuron is the one in the head, the lower motor neuron is the one in the spinal cord. Now, I'm pointing this out because that terminology is actually kind of important to understanding certain neurological issues. The upper motor neuron is basically the source of conscious instructions to move your body. The lower motor neuron is the neuron of the motor unit that we started talking about in the musculature. Okay. So when you are making a voluntary movement, your brain says, let's move stuff, and it tells the motor unit to do something. But sometimes the brain's not involved. So if you are just touching a hot stove and withdrawing your hand, that's completely reflexive. And in that case, the sensory neuron bringing information in, instead of the axon going up to the brain, the axon will just go directly to the lower motor neuron and tell that to fire off so that you contract the muscle and pull your hand away from the heat source. That's just reflex. So lower motor neurons can be part of involuntary reflexive kinds of movements because they just are what control muscle contraction. And sometimes a reflex like that is going to control muscle contraction. Whereas any voluntary movement is going to require an upper motor neuron to be involved. Clinically, there's two sets of movement disorders, essentially. Those that affect upper motor neurons and those that affect lower motor neurons. And I'll actually make sure that I have this straight when I say it when we get to the neurological stuff at the end of the semester, but I want to point it out here. I'm not going to say the names of any of the diseases because I usually get them messed up and I want to double check it before I say it. But there are a number of muscle movement disorders that are really about the voluntary control of muscles, and those are upper motor neuron diseases. And one hallmark of upper motor neuron diseases is the intact presence of reflexes, which means reflexes don't involve upper motor neurons. So if a reflex works, but somebody can't move parts of their body voluntarily, then that's an upper motor neuron problem, which might be in the brain or higher up in the spinal cord or brainstem. When somebody can't reflexively move, or the muscle fibers are flaccid, that they have no inherent tone or contraction to them, that's a lower motor neuron disease. And I can give you a good example of that. Just anything that cuts through a nerve to disconnect the neuron from its target. So if you have some sort of wound that severs a nerve somewhere in your body, then the muscles from that injury out aren't going to get any input from the lower motor neurons, and they'll be flaccid and there will be no reflexes there. That sort of illustrates lower motor neurons. When we're talking about the neurology stuff at the end of the semester, I'll give you some better examples. For example, ALS, or Lou Gehrig's disease, is a movement disorder. And the reason I'm not saying which is I can never remember whether it's an upper or lower motor neuron disease. So I'll address those kinds of things when we're talking about neurology. But I want to point this out because it's in this figure, and we'll come back to that with the neurology stuff at the end. The other thing that I want to talk about, which is sort of the reason for this picture, is to introduce to you why there's signaling in the nervous system. Now, we already kind of talked about this in terms of controlling muscles, but we're going to talk about it in a broader sense, so to speak. The arrows that are drawn in running along the axons here are suggesting the movement of information. And that information is carried through this electrochemical signaling that we call the action potential. And the focus of what we're going to start talking about today and continue on Tuesday is how that electrochemical signaling works. It all happens in the membrane of the cell. And so what we're going to see is a change in electrical properties of the membrane. At the site where the sensory stimulus is activating the endings in the skin, that change in membrane properties is called a generator potential. It generates the signal. And then once it's traveling along the axon, it's called an action potential, which is going to travel all the way along here until it gets to the end of the axon here. The action potential, when it arrives at the synapse, causes the release of neurotransmitter. There is no electrical jump from one to the other. There's not a spark between two cells. Rather, the electrical signal ends at the end of the axon, causes the release of neurotransmitters, which go across the synapse and initiate a new action potential on the other side. Now, I said that there was a generator potential down where the stimulus initially affects all of this. When we get to a synapse, we have something kind of like a receptor potential, which is called a graded potential or a postsynaptic potential. And that will cause the action potential to start in the next cell and move on to the next connection where we have release of neurotransmitter, a new postsynaptic potential, and hopefully a new action potential that travels up there. Okay. And we can say the same thing on the red line going down. It's just, there's an initiation of a signal. When it's from sensory stimuli on nerve endings in the skin like that, it's called a generator receptor. Generator potential to generate things. And then at each synapse, it's called a graded potential or a postsynaptic potential. And then we have the action potentials running along the way. So, I'm going to get back to those terms in a second because I want to define them in a little bit more detail for you. But I just want to show you here in the book, this just highlighting where the sensory receptors are that are, in this example, being exposed to this water stream. And then here's the suggestion of what the motor response is doing. And so the hand is moving to change the water temperature, which is a pretty good representation of trying to present that idea. It's still the left hand that they've drawn in here rather than the right hand. But that technically could be solved just by changing up the picture so that it's showing the right side connected to this hand or something like that. But, and also, I would say, generally speaking, well, I guess I can't really say that, but I would normally think that you'd use two hands. You'd feel the water temperature with one hand and turn the knob with the other, which is a much more complex circuit and really not what I wanted to get into with this example. But anyway, any way you cut it, I don't think it's a great example, and I really need to find a different one to explain things. So now we want to talk about membranes, and I call this excitable membranes, which is a general term for this. I'm going to get out of the presentation for a little while to start talking about those terms I just introduced. But I want to show you where we're going with this. We're going to talk about how the membranes of cells play a role in this electrical signaling. Now, this is going to be presented in terms of the cell membrane of neurons because we're in the nervous tissue chapter. But actually, what we're talking about here applies to the membranes of muscle cells. And actually, there are other cells in the body which are beyond the scope of this class that also have electrical properties that work on the same concepts. But for the sake of what we're covering here in AP1, we're really mostly concerned with neuron membrane excitation and muscle membrane excitation. So that's what we're going to concentrate on. And because of work in this particular chapter, all of the examples that are drawn are really actually going to be of neurons, but they apply to muscles as well. So let me get out of the presentation here for a little bit because I want to come back to these terms that I was starting to introduce. Now, a term I didn't just introduce to you, but is really the sort of granddaddy of all of these terms, is membrane potential. And we got an issue back here?
No.
Okay. This is excitable membranes, and we're talking about the electrical properties of these membranes. And the electrical properties are described as potentials. The word potential is used sort of in the sense of potential energy versus kinetic energy. We're not going to really talk about it in those terms, but that's why the word is potential. Now, a definition of the membrane potential is the distribution of charge across the membrane. This is measured in millivolts, which is abbreviated, of course, lowercase m, capital V. And we'll eventually start talking about some real numbers with this, but this sort of starts to explain what it is. Now, charge in a physiological system is carried by charged particles or ions. In an electrical system like what's controlling the lights in this room, the electrical charge is based on the electrons that are traveling through a circuit, which is actually a little oversimplified way of talking about electrical activity, electricity. But when we're talking about electricity, like controlling lights, the charge is based on electrons. Okay. In physiological systems, the charge is based on ions. Now, ions are atoms that have more or fewer electrons than protons. And so they have a charge based on the numbers of protons and neutrons that they have. They can be positively charged and they can be negatively charged. Electrons, the basis of electricity controlling these lights, are by definition negatively charged. Okay? So don't get hung up on the particular type of charge in that comparison. Electrons are negative and the ions we're talking about are positive. That's not important. It's just that there is a charge. That's the point. Now, I have a object demonstration here, which I'm having a little trouble pulling out of the box. Oh, that's a shame. Where'd it go? Okay, I don't know what happened to it. It's probably in my cart somewhere, but what I would be holding up in front of you right now is a AA battery. So pretend that's what's between my fingers. And the reason I'm showing you a AA battery is because one way of considering what's happening with the membrane is that the membrane is a battery. Okay? It's storing charge. And so the idea of calling it a potential, as in potential energy, fits with that battery description. A battery is stored energy that can be used to make the Energizer Bunny do its thing or whatever. So the battery, the stored energy is potential, and then when it is used to power an electronic circuit, then it does some work, which is kinetic energy. So the membrane acts as a battery in that sense. And so when we say that there's a distribution of charge across the membrane, and that's measured in millivolts, that's just the charge of the battery, so to speak. So again, pretending that I'm holding a AA battery in my fingers, what charge is associated with that type of battery? How much charge is stored in an AA battery? Every so often, I like to ask questions to make you realize that you don't pay attention to the world around you. Every battery says on it what its charge is, but we don't actually care about that value because all we care about is that we put it in our toy and the toy works. Now, I do actually have another battery in my hand, which I could have started with, but I wanted to point out the omission in your paying attention to the world around you. With that. Right. So yeah, this is a 9-volt battery, okay? And it's in the name. Did I say that the AA battery is 1.5 volts? I didn't say that. Okay, yeah, so that's what it is. So if you look at on this brand of battery that happens to have a copper top to it. They usually write the voltage of the battery in the copper part. And this one, of course, says 9 volts. And if it was a AA battery that I was showing you, you would see that it says 1.5 volts. Actually, AA batteries and AAA batteries, even though one's smaller than the other, actually have the same charge. They're both 1.5 volts batteries. Now, I'm using batteries to suggest that the membrane is basically like a battery, and also to give you a sense of what millivolts are. Okay. Now, the prefix milli in a unit of measurement means what? Or how many millimeters are there in a meter? Right, 1,000. So 1 millivolt is 1/1000th of a volt, which means that the 9-volt battery could also be thought of as a 9,000 millivolt battery. Or the 1.5 volt AA or AAA battery is a 1,500 millivolt battery. Now I point that out because the fact that we're measuring this in millivolts is because the charge that is stored up across the membrane is relatively small. It's in the neighborhood of 100 millivolts. Well, yeah, we'll say in the neighborhood of 100 millivolts. So that amount of electricity at any one time isn't a whole lot. Would anybody like to make the next demonstration for me and stick something metal into the socket over there? Okay. How many millivolts of electricity would you be exposed to if you did that?
Enough to kill you. Like all of them?
Not enough to kill you, trust me.
No.
What?
All of them.
All of them. Okay, so again, let's see if people know about the world around them in any detail. What is the voltage of your average electrical outlet?
120.
Yeah, usually about 120, sometimes 110, but which would mean in millivolts it'd be 120,000 millivolts. Okay. Now, I actually do have some personal experience with this. It will not kill you unless you enjoy it for too long. When I was a kid, I was unplugging a television or something like that, and I grabbed the plug, and I actually got my fingers onto the post of the plug, so I got a little jolt of 110 or 120, whatever it was where we were living. So yeah, it's not fun, but it's not gonna kill you unless you put a fork in there and hold on to it for a while. So, yeah, so nobody wants to do that demonstration, which is a good thing. But yeah, that's a lot of electricity. What's going on in our body at the cellular level is very, very little. Okay. So, then the next thing I want to introduce is what's called the resting potential. And this is the actual value of the membrane potential at rest. Okay? So when the cell's not doing anything, we can measure the value of the charge across that membrane, and that's what we call the resting potential. Now, there's some variation in the numbers of what I'm talking about here, depending on the exact cell that we're looking at. But we're going to work with a set of numbers that's generally accepted to be the most common values. And again, we're also going to pretend all of this applies to muscle cells. In truth, the values that we're going to talk about in neurons, while they're not actually universal for all neurons, definitely are not the values that are actually in muscle cells. But they're in the right neighborhood. It's fine for us to stick with just one set of values. So the membrane potential at rest stores a charge of -70 millivolts. Now, the fact that this charge is negative just applies to the fact that, applies to the fact of where we're making the recording. So let me just jump over to the presentation real quick and show you, whoops, not that picture. Show you this picture right here. These are pictures out of another book, I think. So I will eventually put pictures from our book into the presentation, but this is fine for what it's showing us. So we have here the membrane, and we can measure the charge across that membrane with a device called a voltmeter. And the way a voltmeter works is you have two electrodes. You put one on the inside of the cell and one on the outside of the cell and compare the charge across them. So this electrode here is actually a wire that goes through a little glass micropipette or glass needle, so that the wire is essentially inside the cell and not outside the cell. Now, for this picture to be drawn really appropriately, the red recording microelectrode needle should extend outside of the extracellular fluid. Okay. Now, of course, the scale that we're seeing drawn in this picture is completely unrealistic. To do this, you would have a Petri dish with a cell in it, and you wouldn't be able to see the cell because it's microscopic in size, so you'd be looking at it with the microscope. And you would have a glass needle, which you could see would probably be a few centimeters to 6 or 7 centimeters long. And the pointy end, which would get down to a microscopic thickness, would go into the cell. And then the other end, which is wider, would be sticking out of the Petri dish completely. So it would be nowhere in the fluid that the cell is existing, the extracellular fluid. It would pierce the cell membrane and be poking inside the cell, and then the other end of it would be way outside the Petri dish. And so the wire that we see coming out of the voltmeter going into that microelectrode needle would essentially be going inside the cell and completely avoiding the outside of the cell. The other electrode would be sitting in that extracellular fluid, and it's called the reference electrode because we're comparing what is on the outside of the cell to what's on the inside of the cell. Now, we could set this up making the recording electrode the reference one, which would just mean we'd flip all of the things backwards. But convention is we're comparing the inside to the outside. Now, when we do that, we measure the charge at rest, we get negative 70 millivolts. Okay. All that means is that the inside of the cell is 70 millivolts negative, or 70 millivolts below the charge outside the cell. Okay. We're basically assuming outside the cell is 0, and so inside 70 less than that. It could, in fact, be that the outside of the cell is 163 millivolts in some absolute sense. And so inside would just be 93 millivolts. Okay. Those are both positive values, absolute charges in those locations, but the inside, wait, did I do that right? Yeah. The inside is 70 millivolts less than the outside. So comparing inside to outside, it's just 70 millivolts more negative than outside. That's where the value comes from. In this picture, they show negative signs along the inside surface of the membrane and positive signs along the outside surface. And that's where the charges are. Now, I said earlier that charge, or the electricity, is represented by ions. So what that means essentially is that there are positive ions on the outside of the membrane, right next to the outside of the membrane, and there are negative ions right on the inside. And we'll get to explaining what that is in more detail later on. But at this point, we just have the negative and positive signs to represent that. So that's the resting potential. The other important term that we use is reaction potential, which is a little bit more complex to define. It is what we've called the nerve impulse for an electrical signal. But what it is it's a series of changes in membrane potential over approximately 3 milliseconds. That's actually the first time I've technically defined it that exact way, but I like that. So, so that means over approximately 3 milliseconds, the membrane potential changes from the resting potential to a positively charged peak. And the difference between the two is the magnitude or size of that signal. Now, I'm typing this in, defining it kind of off the cuff, although I'm always defining this semester, so it's not like I'm making it up. But so let me make sure that you understand what I'm saying here. Okay, so a cell is sitting there at resting potential. Over about 3 milliseconds, the charge distributed across the membrane changes, going from that resting potential value, which is negative 70 millivolts, meaning the inside is 70 millivolts more negative than the outside, to a peak. And when I say peak, I'm talking about we can draw a curve on a graph paper showing the instantaneous changes in the charge that we're measuring. And it'll hit the top of its peak, which is actually going to be a positive charge. So inside the cell will become positive relative to outside the cell. Earlier I said we're working with values in the neighborhood of 100 millivolts compared to the 9,000 or 1,500 millivolt batteries we were just talking about. And this is where that 100 millivolts really comes in. The action potential, using the numbers we're going to use, changes across 100 millivolts, which means at rest it's negative 70 millivolts inside the cell compared to outside. And at its peak, it's positive 30 millivolts inside relative to outside. Okay, so that 100 millivolt change across the membrane is the basis of our nerve impulse or the electrical signal that we've been talking about. And really a lot of what we're going to do with the rest of this part, well actually mostly on Tuesday, is to explain how that happens. The exact changes in the membrane, in the cell, that are occurring during that 3 millisecond window that's the action potential. Now, I want to define a couple of other terms that came up earlier as I was showing you the example there. One is the graded potential, and this is a change in membrane potential directly related to the strength of a stimulus, which is to say, very cold water will cause a big change, and just slightly cold water will cause a little change. It might be, the difference between 100 millivolts. Well, no, it's not going to be 100 millivolts. It might be the difference between 20 millivolt change in potential versus a 2 millivolt change in potential. It's graded. It's directly linked to how strong the stimulus is. Now, the first potential I mentioned, I said, was a generator potential. And this is a graded potential in a sensory neuron's dendrites. Okay. So when the dendrites of a neuron are directly interacting with a stimulus, like the cold water or the chemical molecules that you smell when you inhale the fragrance of a rose or something like that, whatever those are. That stimulus, and there are a whole bunch of different possible things that can be stimuli, when they interact with the dendrites of that sensory neuron, the thing that's directly interacting with the stimulus, it produces a graded potential, which is called a generated potential in that situation. Okay? It's a type of graded potential. The stronger the stimulus, the greater the change. But when we're talking about one of those physical stimuli from the environment around us, the sensory neuron that's interacting with it makes a generated potential. Then another thing that I want to mention related to that is called a receptor potential, which is a graded potential in a specialized sensory cell. Okay. Now, the examples I just gave you for generator potentials were a temperature receptor in your skin or the odor receptor in your nose. And with the skin, it can be anything: pressure, vibration, temperature, pain, any of those things create generated potentials because those are sensory neurons. A receptor potential is the same thing, but in a different type of cell, which is a specialized sensory receptor or specialized sensory cell. And a good example of that would be photoreceptors in your retina, or the taste receptors in your tongue. Because those are not neurons. Those are specialized cells that interact with stimuli in a very specific location, and they change activity where they connect to a sensory neuron, but it's not the sensory neuron itself. Let me use chemical stimuli for an example to make the point here. A smell sensory neuron will generate a generator potential because it directly binds to the odor molecule that you inhale. A taste bud will make or the cells in a taste bud will make a receptor potential because the cells in the taste bud aren't neurons. They are specialized receptor cells. So it's just depending on what type of cell is interacting with the stimulus. There are those two different types. And then the last term I want to introduce here is a post-synaptic potential. Now, as I started to type that in, I hyphenated post-synaptic, and then I erased it. When I was a student, pretty much post-synaptic was a hyphenated term. But I'm really trying to get used to leaving the hyphen out because more often, and probably more grammatically correct, it's not hyphenated. Post is a prefix, synaptic is the root word. So post-synaptic really shouldn't have a prefix in it. It just, back when I was a student, the convention was to put a hyphen in it. So if you see me hyphenating it there, just remember that's because I'm old and I forget to do those sorts of things. But a postsynaptic potential is a graded potential at or in a target cell at a synapse. So the interneurons that I was pointing out, they don't have generator potentials, or I didn't talk about them at the time, but they don't have receptor potentials either because they're not directly interacting with the sensory input. Instead, they're getting an input from another cell, another neuron. And so they make postsynaptic potentials, which is another type of graded potential. So if you put your hand under cold water and it's cold enough to generate an action potential in that sensory neuron, that action potential will go up to your brainstem, for instance, and release neurotransmitters at that synapse. Now, if it's actually a fairly weakly cold stimulus, at that synapse, probably not enough neurotransmitters will be released. And the postsynaptic potential will be very weak. if it's a very, very cold stimulus in the hand, when it gets up to the brainstem, there will be a lot of neurotransmitters released, and that'll cause a stronger graded potential or postsynaptic potential. So all three of these things are graded potentials. They're dependent on the stimulus. Generator and receptor potentials are usually dependent on the physical stimulus in the environment around us. And postsynaptic potentials, the stimulus that they're dependent on is the amount of neurotransmitter released at the synapse. Okay. Now, I'm making this point because the actual thing causing the graded potential is different depending on what type of cell we're talking about. But really, they're all the same kind of change in the membrane. It's how much the membrane potential changes, 20 versus 2 millivolts, something like that, depends on how strong the stimulus is. So I just wanted to introduce those terms. Now, I wanted to introduce these terms and define them for you. You have them in your notes. We're going to be using these terms as we describe the electrical signaling and activity for neurons and that sort of stuff. So I wanted you to have all of these at your disposal. And actually, I'll probably introduce a couple of other definitions that'll come up later on, but they won't make as much, well, like these probably make any sense to you now anyways, but a couple of other things I want to introduce won't make any sense to you at this point because I have to start talking about the membrane before we get there. So this is not a complete list of definitions, but these are a good set of definitions that we'll use to talk about these things. So what we're going to do is, again, talk about excitable membranes. And in that, we're going to talk about how, oh, actually, now that I look at all these pictures, these are the pictures of the block. So we'll talk about, sorry? We'll talk about what allows things to move across the membrane, how we can change the voltage or the charge measured at the membrane. And then we'll talk about how that leads into making these electrical signals. Now, if you're sitting there going, wow, it really sounds like he's wrapping things up, that's because I am. I could certainly spend 10 more minutes talking about this stuff, but this is a starting into a new subject, which really requires more than 10 minutes to get through completely. So I don't want to start it now because then on Tuesday, I just have to start it all over again anyways. So I'll save this for Tuesday and we'll continue talking about this stuff. And hopefully get to explaining what the action potential is. Monday, of course, we have a lab, and we'll be looking at the anatomy of the brain mostly to get a sense of what's going on. So like, last week, we were looking at the cells that make up nervous tissue. In the next lab, we're going to be looking at the organs that make up the nervous system in more detail. So we'll pick up with that. Otherwise, that's all I have for today. Have a nice weekend, and I'll see you on Monday.